The biggest mistake people make about programmable biology is thinking the scarce resource is ideas. It is not. The scarce resource is fast, reliable validation. In the discovery economy, advantage goes to whoever turns a design into a decision-grade biological result again and again under clearly stated conditions. Start with what the product really is. It is not a paper. It is not a model. It is a reproducible mapping from a design specification to a measured functional outcome. That sounds abstract, but it is the only unit that compounds. If you can produce those mappings faster, you can learn faster. If you can learn faster, you can allocate capital with less guesswork. Modern biology already has a shared coordinate system. That became real with the initial sequencing and analysis of the human genome in 2001. Once you have a reference, you can anchor measurements, compare results across groups, and build tool chains that assume a common language. Later, that substrate gained statistical weight. In 2020, researchers quantified a mutational constraint spectrum using data from 141,456 humans. Practically, that gives you priors. It helps answer, probabilistically, which kinds of changes are likely tolerated and which are likely harmful. For strategy leaders, that is a shift from edit and hope toward edit with informed risk gates. Now consider the edit tools themselves. A key expansion happened in 2016 when base editing demonstrated programmable single-base conversion without double-stranded DNA cleavage. That matters because it adds a finer primitive for design. It can also reduce certain damage pathways associated with cuts. Still, even with better primitives, the hard part remains, predicting phenotype from genotype across different cell types and contexts. The same edit can behave differently depending on where it lands, what else is happening in the cell, and how the system responds over time. Computation helps, but it does not replace reality. A clean example is Swiss ADME, released in 2017, which standardized pre-wet lab screening for pharmacokinetics and drug likeness. Tools like that compress candidate evaluation time. They reduce the wet lab burden by filtering out obvious losers early. Yet they must be audited against outcomes because standardized predictors can also standardize blind spots. If your organization scales triage faster than it scales verification, you can end up confidently wrong at high throughput. Delivery is where many elegant designs go to die. A designed payload is not a delivered payload, and delivery is not a side quest. It is a first-class engineering discipline. A 2020 synthesis of precision nanoparticle drug delivery captured this plainly. Delivery constraints often dominate what you can achieve in organisms. Tissue specificity, repeat dosing, manufacturability, immunogenicity, toxicity, all of these can set the ceiling on impact. When delivery is the bottleneck, investing only in payload design is like optimizing software while ignoring the network. Platform stories show both the promise and the boundaries of fast iteration. mRNA vaccines were characterized in 2018 as a platform modality with engineering properties that can enable faster iteration than traditional paradigms. Adenoviral vectors provide another anchor. In 2020, Phase 1 TAPU safety and immunogenicity results were reported for ChAdOx1 nCoV19, demonstrating an accelerated clinical evaluation pipeline for a platform vector. The point is not that regulation disappears. It is that certain platform properties, modular manufacturing, standardized analytics, predefined safety packages can compress translation time in bounded regimes. Even then, measurement quality often determines the real learning rate. Assay noise, batch effects, and inconsistent baselines can destroy iteration returns. You can build at high speed and still learn slowly if readouts are unstable. That is why the high-leverage work is often boring work. Standardizing assays, tracking coefficients of variation, defining acceptance thresholds, and keeping a unified experiment ledger that links every design choice to every readout. Without that, you do not have an iteration engine. You have activity. Context dependence is the quiet limit that keeps programmable life from becoming a simplistic metaphor. A 2020 review of gut microbiota and metabolic health highlights nonlinearity and ecological feedback. Change the diet. The microbes shift. Shift the microbes, the metabolites change. Those changes loop back into host physiology. For anyone planning interventions, the question becomes controllability and reversibility, not just efficacy in a controlled snapshot. Finally, remember that not all discovery spaces are well parameterized. Natural products, surveyed in 2021, remain chemically diverse and difficult to model. That means exploration still matters. In many domains, the best pipeline is not purely model-guided design. It is a disciplined allocation between computational guidance and empirical search, with tight feedback loops that update priors.